This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 195, recorded on August 10th, 2012. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the weekly discussion show all about viruses. Joining me today, after a decade of absence, Dickson Despommier.
That's not fair.
I'm so sorry that I bother you, but I can't resist.
Vincent.
Welcome back, Dickson.
Well, thank you, Vincent. It's a pleasure to be back after these decades of absence.
It feels like forever.
It does.
But I'm glad you could make it.
No, it really does feel like a long time, and it has been.
Dickson's right here in the TWiV studio. I can see him.
That's true.
Where it is pretty warm, isn't it?
It's not so bad right now, but it was warmer yesterday.
Well, they shut the air off here.
Yeah.
That's the problem. Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
With a intact router and good internet.
Yes. Yeah, the thunderstorm just rolled in about an hour ago, but I managed to stay online.
It's the one we see here, Dickson, right?
I think, it's probably a different one, but related to the same system.
Yes.
Thunderstorms.
Yes. Also joining us from North Central Florida, Rich Condit.
Hi, fellas.
Hey there.
Hey, Rich.
How's everybody?
Good. How's the
Good.
weather?
31 Celsius, that is 88 degrees Fahrenheit, and mostly sunny skies. Not too humid for Florida.
Nice.
It's very nice.
Right
Nice
here
day.
in New York, Dickson. Do you know what the temperature is?
I would guess it's about 81.
It's 21 degrees centigrade, or Celsius, I should say.
21.
Yeah.
That's almost room temperature.
Yeah, can that be right?
It's a little below room temperature. That's probably the storm system rolling in.
I'll bet, because it's really hot in here.
Yeah,
It
because
is.
it's here. The temperature plummeted. It's in the mid 70s
You should
now.
open a window.
It's supposed to be 27. Yeah, windows. What's a window?
That's right.
All right, we also have a special guest today from, this has got to be the best department or center name that I've come across, the Center for Life in Extreme Environments, which could be here in New York City, right? And the department, the biology department at Portland State University, Ken Stedman.
Okay.
Hi, everybody.
How you doing there?
Okay.
Pretty good. Actually, today the weather's not terribly extreme in Portland. It's pretty normal for us. Overcast skies, but
Right.
about 22, 23 degrees Celsius. Should be clearing up in about an hour or so. No rain. Actually, we could use some here. It was kind of funny. Last weekend, I was in San Diego, and it was about 20 degrees colder there than it was here. We had a heat wave come through.
Wow.
So you're in Portland.
In Portland.
Downtown
exactly, yes.
Portland, right?
Downtown Portland, yeah. We're literally right downtown. It's a very urban campus here, which is great. Portland's an absolutely wonderful place.
Yeah, we were there. I was there with my family 2
I
years
agree.
ago.
I have a good friend there who owns a restaurant. Ken, you might want to go to it.
Which one?
It's called Andina.
Oh, Andina's fabulous.
Okay.
Can I get a discount?
Yeah.
Sure. Just tell them that Dick Despommier sent you, and they'll kick you right out.
There you go.
Downtown in Portland, there's
Peter Platt runs the restaurant.
There's a little Japanese garden, right?
Yeah.
We went there. That was really cool.
No, it's a great city. It's a
beautiful.
great city.
Really nice.
It's really nice.
Well, the reason we have Ken here is we're going to talk about his paper today, which came out in April? Is that right?
Mm-hmm. Yeah, exactly.
In Biology Direct,
Correct.
which I'm not familiar with. Were you previously familiar with this journal?
Yeah, so Biology Direct is a journal, completely online open access, which was founded by a colleague of mine, Eugene Koonin. And
okay.
it's got a really fascinating editorial system, which I'd sort of heard about from him, and I thought we'd give it a try. And it's a completely open review process, and so the reviews actually end up getting published together with the manuscript
Interesting.
and the responses to the reviews. So
That's really scary.
Very scary.
yeah, and so it's the other
great
thing is, so
idea.
basically, because of that, when you submit an article to Biology Direct, if they accept it for review, it basically says it's going to be accepted.
Right.
So it's the editor makes that call very early on in the process. And what you can do, if you like, just again, because it's a completely open review process, you get back your reviews, and you can just say, well, blank you to all the reviewers.
That's where I went to
because
school.
their reviews will be there.
Yeah.
And so you've got, your manuscript, you've got their responses to your manuscript. Now, in our case, the reviewers were actually extremely helpful and I think greatly improved our manuscript. But it's a very interesting and different way to go about doing things. So
probably makes people tone down the reviews, right? Knowing it's going to be public.
Potentially, although
Do they have to sign them?
It's pretty direct.
Yeah.
They're all
They have
signed.
to sign. They're all signed. That's great. I like that a lot.
When I write reviews anyway, I think everything I ever write would be publishable.
Yeah.
I'm never really, nasty or anything like that.
Right.
So,
Yeah, I agree.
and most of the reviews I get are like that too. So.
Is this the first paper you published in this journal, Ken?
Yeah, it's the first one we published in that journal. It's but I'm seriously thinking about publishing some other things in there as well. It was really quite nice. Now, the manuscript was actually declined by another journal, which will remain nameless.
Oikos or something trivial like that.
Yeah. One of the other reasons we decided to go to Biology Direct is they have a pretty quick turnaround time.
Yeah.
And we were actually rather concerned that some of the results that we had in this paper could have been easily scooped by someone else. So we really wanted to get it up quickly.
So here's the history. You had actually sent this to me a long time ago, and it was in the typical long TWiV email queue, and I didn't get to it.
Yeah, I was familiar with this.
And
somehow.
then
maybe because of your blog.
I think a listener sent it in then again. I think our friend in Portugal. And then that caught my eye for some reason. I blogged about it, and then I found your email, and we got in touch. And this, interestingly, on my blog, usually only XMRV and H5N1 generates any discussion. But this paper actually generated quite a bit of interesting discussion. So I figured we should tackle it here on TWiV. Before we do that, though, I want to find out your history. Where'd you come from?
Well, that's. Do you want the long story or the short story?
Depends how good it is, I guess.
If you start with Old Divey Gorge, we're going to cut you off.
Yeah. Well, so
Haploid.
There you go. Exactly. The haploid to diploid happened in the United Kingdom. So I was born in England, British parents, and then my parents came to the U.S. purely temporarily as postdocs 44 years ago. They're still here.
Wow.
They're no longer postdocs, fortunately. And curiously enough,
They're now retired postdocs, right?
Exactly. The first place that they moved to was Manhattan.
Manhattan, Kansas?
Manhattan, Kansas, exactly. Not Manhattan, New York City.
No.
And so Kansas State University. My father
Just
was
going
now an
through it.
emeritus chemistry professor.
Nice.
And I bounced around the U.S. a little bit, ended up growing up in Michigan, and then went and did my undergraduate degree in chemical engineering, rather like one of your previous guests a couple of TWiVs ago, at Stanford. And then I took the really long route from Stanford to Berkeley via Switzerland for 3 years, and worked in biotech there for a couple of years, and decided that molecular biology was really much more exciting, at least to me, than chemical engineering. And then got my PhD in Sidney Kustu's lab at the University of California, Berkeley, working on transcriptional regulation in salmonella. And so I got really excited about transcriptional regulation doing that work.
That was UC where? Berkeley?
Berkeley, yes.
Okay.
And from there, I actually then I'd found out, partly because I was TAing the course that Sidney Kustu was teaching on just general microbiology. I heard about the archaea, and I got really excited about the archaea as this great model system sort of between bacteria and eukaryotes, and particularly at the transcriptional level. So when I was looking for a postdoc, I basically wanted to do a postdoc doing transcriptional regulation, but not in bacteria, but in archaea. And so I looked around, and at that point, there were basically 4 labs in the world that were working on transcriptional regulation in archaea. There was 2 labs in Germany, one in the UK and one at Ohio State. And so I went and interviewed at all 4 labs and ended up going and working with Wolfram Zillig, who's really the pioneer in the field of both archaeal transcription, but what I didn't realize at the time was also in viruses of archaea. So I went
And where was that?
That was at Martins Reeds, the Max Planck Institute for Biochemistry, about 100 meters outside of Munich. So it's basically Munich. Fortunately, having worked in Switzerland, I spoke German, so it was easy for me to be able to go and work there.
Yeah.
And just basically when I arrived, there's a wonderful story that I like to tell people. I get there, and the first half hour he spends telling me about how horrible the German system is, and it's forcing him to retire, and he's so active, and all of this other stuff. And I just think to myself, I really need to work for this guy. He's just too much fun not to work. And I get there to postdoc. He says, "Well, I'm sorry, I'm not going to shake your hand because I was just spearfishing off of Baja and shot myself through the hand."
Oh, Lord.
Sounds like you would be doing that, Dickson.
I don't spearfish. No, it's
So I ended up working in his lab, and the idea really there was to try and combine what I'd done working in bacteria into an archaeal system, and so it was really a combination of really good biochemistry, but also some genetics. And the big problem with archaea at the time, and still to some extent is the case, is there's very little genetic tools available in archaea.
Sure.
Of course, what are viruses? Viruses are absolutely wonderful genetic tools. So basically ended up using a virus, which they've been characterized in the lab, in fact, for quite a long time. I think the first or second virus of archaea that had ever been discovered in 1982. This is the SSV virus, kind of looks like little lemon particles.
Yeah, it's in our textbook. Yep.
It's just it's a fascinating virus.
The one that matures outside of the cell?
No, so that's actually a slightly different one.
But is that also an archaeal virus?
And so when I got there, so I did in fact use and set up basically the main outcome of my postdoc was setting up a genetic system using this viral genome. It's a double-stranded DNA, about 15,500 base pairs in size, replicates as an episomal plasmid. So it's actually really quite well behaved. And so we, in fact, use that virus for a genetic system here, and we've now set up a reverse genetic system as well. So it's, I think, becoming one of the sort of model viruses of archaea. But I was really interested in the transcription. So transcription, there are people in my lab who are still working on transcription. But I got really excited about the virus because they were just so different. You look at the virus genome, this SSV virus genome, getting 15 kb, it's about 35, 36 open reading frames in it. You compare those open reading frames to things that are in the database, one and only one of those has any kind of match.
It's great. Yeah. So this is our first archaeobiologist on TWiV, right?
I guess.
No one's going to say no, unless Dallin and Rich are gone.
I don't
I'm here.
recall.
So this SSB virus, it's double-stranded DNA, linear,
Circular.
Yeah. And the bug that
circle.
it grows on is not an extremophile, or is it?
It is. So the bug that we work on is called Sulfolobus sulfatericus and grows optimally at 80 degrees Celsius and pH 3.
Nice.
So how do you manage that?
Wow. Basically treat it kind of like E. coli. So we take our media, we pH it 3 with sulfuric acid, we heat it up.
Lord, that's like growing on Venus.
It grows great. And in fact, it's really nice because you don't have to worry too much about bacterial contamination.
Or anything else, for that matter.
About cross-contamination from some of the other isolates. So that actually has been a little bit of an issue. But at least as far as the bacteria is concerned, no, we don't have too many worries about that. And it was also great to be working on this particular virus because first you have to find an appropriate environment. How do you find something that's 70 degrees Celsius plus and pHs of 3? Turns out it's volcanic hot springs. And so I had no idea when I got into this field that I'm actually doing a little work. That,
So-
for me,
So I mean, in the lab, you at 80 degrees. I mean, how do you keep these things warm, and how do you keep them heated? I'm trying to imagine a shaking platform or something like that that's at 80 degrees and where stuff won't evaporate but is still aerated. How do you manage this?
So one thing to bear in mind, these are actually microaerophiles. They don't need very much air. And if you think about 80 degrees Celsius, the solubility of oxygen is pretty low at that point.
Very.
Don't have to shake them very much, but we do. It turns out that these are, in fact, aerobic, probably again, aerophilic microorganisms. We put them in a regular New Brunswick shaker. And I have a long-necked Erlenmeyer flask. We either make them ourselves or you can actually buy them. So you actually have a little bit of a reflux column for the actual culture.
Okay.
And so basically it evaporates and then will condense and then come back in. So that works really quite well. The other thing, of course, is your bath fluid. Now, if you use water, it's going to evaporate pretty quickly at 80 degrees. Yeah. So we use polyethylene glycol.
That's great.
400, which is great. Heats up nicely and cleans up with water. You can use oil as well, but it gets really messy really quickly. And you don't make friends with anybody.
Wow, antifreeze.
Is there a plate assay? Can you?
Yeah, we actually can grow them on plates. We use something called Gelrite, also known as phytogel. Plant biologists have been using it for quite a long time. In the presence of divalent cations, it's a Xylan gum. But in the presence of divalent cations, it actually sets up really nicely. And so you can get these plates that are stable at 80 degrees. In fact, some people even use them at 90 degrees Celsius. The problem there actually has to do with the pH. If the pH is too low, the plates have a nasty tendency to melt on you, and it becomes a little.
Do you have to seal them so they don't dry out?
We usually put them in Tupperwares.
Okay.
A little bit of extra
chamber.
water.
Yeah.
And you can get plaques.
We get actually really quite nice plaques. They're turbid plaques. It's a little hard to see sometimes, but they make pretty decent plaques.
I just remember as a graduate student, we tried to grow Bacillus stereothermophilus in the lab, of course, without any clue what we were doing. And I just, I remember one of my fellow graduate students taking a petri dish out of an incubator and opening up, and all of this vapor comes steaming off of it. Weird.
So, Ken, if you don't grow it at 80, say, if you grew it at some lower temperature, what would happen?
So we've actually looked into that quite a bit because we're interested in virus-cell interactions. And turns out that below about 60 degrees, 65 degrees, they don't grow at all.
Wow.
And that's actually really kind of useful because we can use that as a freezing technique. And
relatively speaking.
we can lower the ambient temperature by 40 to 50 degrees Celsius and we're at ice.
Yeah.
So we can basically shut down whatever production of particles just by lowering the temperature.
Yeah.
Wow. So, Ken, can I ask, the original source for your material that you're working with comes from a volcanic, a pond or a puddle or a bubbling mass of quivering jelly? Tell us where you got this stuff from.
This virus is talking
about? Yeah.
So the SSV1 virus was originally isolated again by my postdoctoral advisor, Wolfram Zillig, a place called Bepsen in Japan, Kyushu.
Okay.
And we've actually isolated similar viruses from Kamchatka and which is just amazing. And I was very fortunate to be able to go there. So we isolated some viruses from there.
Right.
And from Yellowstone, from Lassen Volcanic National Park, pretty much anywhere where there's volcanic activity, you'll find sulfolobus, and you'll find in about 5 to 10% of the samples, you'll find these spindle-shaped viruses that are associated with them.
Remarkable.
So, in fact, the host that we use is from Igerelli, which is just outside of Naples, Italy.
Right. That would be Mount Etna, as I recall.
The place, Solfatara.
Vesuvius.
Right next.
Vesuvius, yeah.
In fact, in antiquity, this was known as a place where you would go and talk to the sages who had been breathing in a little bit too much of the volcanic fumes, hydrogen sulfide and the sulfur dioxide, and get your oracle from there. And so,
right.
So then that brings us to your current paper. It's called "A Novel Virus Genome Discovered in an Extreme Environment Suggests Recombination Between Unrelated Groups of RNA and DNA Viruses." So this involves going out in the field and collecting. So tell us a bit about why you're doing that.
Yeah, so this particular project is part of an NSF-funded microbial observatory program, which I'm collaborating with two groups in Northern California, Patty Suring and Mark Wilson, who are at Humboldt State University, and Gordon Wolf, who's at California State University at Chico. What we're trying to do is understand the ecology of an acidic hot lake. And this is actually slightly lower temperature than Sulfolobus likes. It's only 50 degrees Celsius. The pH is about 2.5.
Remarkable.
There are regions of the lake that get up to 95, 96 degrees Celsius, and that's boiling at that altitude because the altitude of the lake is about 6,000 feet.
Oh, yeah.
Wow. And this is in California, the lake, right?
This is in Lassen Volcanic National Park. So it's the southernmost of the Cascade volcanoes. Now, from Portland, if you're lucky, you have a really nice office. You have a view of our local volcanoes.
Right.
And if you follow those all the way down to the south, you get Shasta, and then the volcano beyond that is Mount Lassen. And before one of the local volcanoes blew up, Mount St. Helens, Mount Lassen was actually the last volcano in the continental U.S. to actually have an eruption in 1914. It's a relatively close pace for us to go, but the great thing about this lake is it's a purely microbial ecosystem, and a very simple ecosystem at that. And so what we wanted to do was to analyze the diversity, the dynamics, and really sort of who's there and what are they doing in this simple microbial ecosystem to understand what's going on in that ecosystem, make some nice models to then compare to some other ecosystems as well. Mine was looking at the viruses. Work on viruses in extreme environments. And so what we were doing was basically trying to find the viruses that would infect the microbes that were in the lake, mostly because we think that they're sort of the only, or at least the main predators that are present in the system. People don't usually think of viruses as predators, but in this case, they're probably providing most of the top-down control from an ecological sense. Terms of regulating, and one of the theories that people have in virus ecology is called kill the winner, whereby you have one particular population which explodes, then viruses come in and take out that population. If anything, you have more biodiversity in your system because if anybody gets too numerous, then the viruses will actually infect that and cause those to be, those particular organisms to be reduced, and can actually be in there. So looking at all the viruses that were present.
So tell us what microbes live in this extreme lake? Do we know?
So we have a pretty good handle on that. Most of the work that's been done on the microbes is again by my collaborators, Patty Suring and Mark Wilson at Humboldt State and Gordon Wolf at Chico State. They found a number of really pretty novel, both bacteria and archaea that are present in the lake. In terms of the sequences that you look at that are there, there's some pretty well-known acidophilic microorganisms which are present there. And in fact, Sulfolobus relatives, although very distant relatives of Sulfolobus, which are present in the lake. But one of the reasons that I was really excited about this lake, and this sort of connects to the paper, I'm sure we'll get there eventually.
Don't bet on it. I want to talk to you about Lake Vostok next.
There are stories about that too, but we'll see. The really exciting thing for us is that there are also some microbial eukaryotes that are present in the lake. 50 degrees Celsius is just about
unbelievable
for some of these guys. And so there's acidophilic algae, which can grow conditions. There's some fungi, and really exciting for us, our collaborators, particularly Gordon Wolf now at State, isolated some amoeba.
Wow.
And so we're very interested in these guys and think probably these ukes that are actually being infected by some of these viruses.
How big a lake and how deep?
So, great question. So the lake itself is about 100 meters by 200 meters.
Right.
The depth, before we actually started this project, nobody knew. It's a huge amount of turbid material, which is in the lake. Lots of suspended sediments, which are just typical volcanic hot springs that these very acidic volcanic hot springs. Sure. So one of the things that we did as part of this microbial observatory program is we had the mechanical engineers here at Portland State University build a little boat.
A non-soluble boat.
And this is what graduate students are for, right?
Usually we take undergraduates up there, but none of them wanted to. So, go figure. So basically a remote control boat that we took out on the lake with a sonar unit on it. And since this was a low budget approach, it's actually a fish finder.
Sure.
Put on here.
Fish locator, that's right.
If we found any fish, I'd be extreme. They would not be very live. No. But one of the things that you have because of the volcanic heating which is happening in the lake, there are lots of bubbles that come up in the lake. And those actually show up as fish on the fish finder. So we use the fish finder, and it turns out that the lake is about 10 meters deep, as far as we can tell. But one of the strange things is that you have a sonar reflective layer down at about 10 meters. It's really flat across the whole bottom of the lake. And whether that's a bottom, whether it's sediment, we're not entirely sure about. So we're now retrofitting.
Could it be a thermocline?
It could be a thermocline, could be a chemocline, could be all kinds of interesting things. Of course, we want a sample at that depth. And so we're now retrofitting this boat to have a sampler we can go out and collect it with.
So this strikes me as actually not a huge volume of liquid. I'm wondering what feeds it, keeps it from drying out.
Right, that's a great question, because if you can also do some calculations. A lot of people at the U.S. Geological Survey have done these things. The amount of power needed to actually heat that volume of water to 50 degrees and keep it at 50 degrees year-round, it's 50 degrees Celsius, of course, is about what you need for a small power plant.
Okay.
So it appears that what's happening is there's steam vents below the surface, which are probably what's causing the heating to actually take place. Whether there's any water coming in is not entirely clear. There's some seasonal runoff that happens. You get huge amounts of snow up there in the winter. So there's a lot of runoff that happens. And so basically every year you have sort of a filling of the lake. And until about May, June, there's some outflow. But after, again, May or June, that outflow usually stops up and the lake just slowly evaporates, basically, until the next major snowfall.
Okay.
Do you have any feeling for how long it's been in existence?
Again, a great question. So, as I mentioned before, the last eruption of Lassen Peak was in 1914.
Right.
It turns out that actually there are reports from the Whitney expedition, which was in the 1850s, of a lake in that location that was too hot to touch. So it probably predates at least the last eruption. But again, before that, unfortunately, there's not much in terms of the Native American oral history. We did a, unfortunately, rather good job of wiping out most of the locals. So there's really not much more. So we know at least 1850, but earlier than that's not clear.
So how do you go about sampling this kind of lake for viruses?
So basically,
Very carefully.
You have long poles, and so that's we have these poles, which we attach scoops to one end of, reach them out into the lake, and scoop up lake water to get the viruses. Basically, what we do is we do a series of filtrations. So we get rid of the big stuff, all the cells and most of the sediment, and then take the smaller stuff, anything that's lower than about 0.45 microns, and concentrate that to as small a volume as we can get on a 10 kilodalton or 100 kilodalton membrane.
So you would miss any really big viruses, like Mimi-like viruses, right?
So we in fact tried to get these bigger viruses because we thought that it was quite likely there actually would be some of them in there, particularly the we found there. Yeah. And we found that we had so much bacterial contamination when we didn't do filtration down to 0.2 microns that it really wasn't that useful.
Yeah.
I seem to recall, maybe it's in this video, pictures of. Oh no, it's on your. There's some on your website here of sampling these areas.
Yeah,
so you can see you're working with the big long poles.
So do you do that yourself, Ken, or is it postdocs and students who do that?
I go down there myself. It's for me the first time I actually went out in the field and was able to sort of look at one of these hot springs and think, I'm actually working on something that's in there. That was really kind of an epiphany as far as I was. It's connected. When I was in my PhD, I'm working on things that show up as bands on gels.
Right. Yeah.
As everyone likes to say, molecular biology is about moving invisible amounts of colorless liquid from one tube to another.
Yeah.
So that was great for me. So even people in my lab who aren't working directly on the viruses or these isolations, I try and get them to go out in the field just so they can have that experience.
Right.
I just love it so much. The location is absolutely gorgeous. So we make a family trip out of it. I go down there with my family.
How often do you
pack
go
your
there?
beach chairs?
That's right.
Show up at the lake with towels and flip-flops.
No windsurfing, please.
You've suggested that they go somewhere else to swim.
Right.
So do you go down there often, or is it just a few times a year?
Usually a couple times a year.
Yeah.
It's still about 9 to 10 hours drive from here to get there.
Yeah.
So it's a bit of a challenge. But our collaborators in Chico are only about a couple hours away, so they'll often collect some samples and then send them up to us.
So now you have these samples which are on filters, and you bring them back to your lab, and what do you do with them?
So we do a couple things. One, we screen to see if there's anything in there that will infect our lab strains of hosts. And then that was unfortunately really unsuccessful. And so then we decided to take a step back and do the metagenomics, which is what this paper is about. And this was made possible through funding from the Gordon and Betty Moore Foundation, those of Intel fame, who are very interested in marine microbiology. And turns out that there's some similarities between this lake and potential deep-sea hydrothermal vents. It's hard to get this lake and hard to get samples, but it's a lot harder to get to deep-sea hydrothermal vents and much more expensive. So we got some funding from them to do some sequencing, basically one round of 454 sequencing, which is about 400,000 sequences. So basically we collect the viruses, or the virus-sized particles, I should say, and then isolate all the nucleic acids from them, and then randomly sequence them.
And then,
is there any sort of amplification step in there, or is it just direct sequencing?
So we tried to get enough DNA to be able to go without amplification. We got to 400 liters of the lake water. And my students basically gave up and said, this was a lot of pumping and filtering.
I'll say.
And they said, okay, well, we'd like to do this without amplification, but we are, in fact, going to have to amplify. So we amplified with 529 polymerase, so the rolling circle strand displacement amplification, which is supposed to be pretty random. How random it is open to some questions, and there's been some analysis of that. So we ended up having to do amplification.
So you got a lot of sequence back, and then in your lab you analyze the sequence as well? You guys do bioinformatics?
Yeah, so there's a lot of tools, fortunately, online of 400,000 sequences. I had to go through a lot of graduate students, getting them to all analyze those individually. So there are a number of programs that are available online for doing searches, and we just basically automatically had these searches run. Took a couple of weeks. And basically the searches which we did are what's called T-BLAST-X. So we took all of our nucleotide sequences, we translated those in silico into proteins, and then searched all of those potential proteins or amino acid sequences against all the nucleotide databases that again been translated in all 6 frames into various different proteins. And so that was the analysis that we started with. And one of the nice things about that is we actually then went from about 400,000 sequences down to a couple of thousand sequences that actually had what looked like interesting matches to viruses.
So what's the total diversity from your sequence? Can you estimate that?
We can guess. It's really hard to say. There are a number of programs and mathematical formulae to try and get an idea on exactly sort of how many different families of virus genome, whatever else you have in there. And it looks like, again, running some of these programs, we've got pretty low diversity in the system, which is one of the reasons that we wanted to look for it in the first place, and in fact probably why we found this particular genome that we talk about in the rest of the paper. We're only finding probably on the order of 50 to 100 different virus families in that set of sequences that we know are similar to viruses. The dark matter, the 96% of the sequences which are in there that don't match anything, are probably viral, but we don't have a really good handle on what exactly they're doing. We've done some analyses, and it looks as if we can take a number of these. So these are 400,000 sequences, but a lot of those sequences you can fit together, make contiguous sequences out of them, the whole contig approach. And those actually, there's some pretty big contigs in this dark matter. So we're very interested in looking into that.
So
what define big?
All relative, exactly. I think, again, I'd have to go back and check, but we're at about 8,000 base pairs. Okay. And the reads, to go back to those people who are not used to 454, we use the titanium technology, or the Bird Institute, who did the sequencing for us. So we've got about 400 base pair reads, which are
so if you were to take the large particle fraction rather than the small particle fraction, that is the hosts rather than the viruses, and analyze that DNA sample, how many species do you think you're dealing with in that lake?
That's hard to tell, but we're just, in fact, putting together a manuscript again, together with my collaborators at Humboldt and at Chico, and it looks as if at least the dominant species, groups of organisms, are probably on the order of hundreds at most.
Hundreds. And is your assumption that they all have viral parasites?
We don't know.
I mean, has there ever been a bacteria or fungus or algae found that that wasn't susceptible to some of these intracellular fantastic voyagers, so to speak?
Not that I know of.
Yeah, exactly. So you'd expect that each one of these had a different set of parasite-host relationships. It's fascinating. I mean, it sounds like a limited ecological setting, but when you start to break it down in terms of microbial life, it becomes complex again.
But still much less complex than the human gut. So it's, I think, a much more tractable system.
So of the 4% of the sequences where you got a match, do they match up with known hosts in the lake, bacteria, archaea, and so forth?
So those that matched were not great matches. So unfortunately, it's hard to really clearly tie down what they're going to. But most of the matches were actually to known viruses of archaea, things that we'd been working on before. Some of the other people that were in Wolfram Zillig's lab where I did my postdoc, particularly David Rangishvili, who's now at the Institut Pasteur in France. So most of those virus hits were to these viruses of archaea, which is exactly what we were expecting.
Right. So the subject of this paper, how did that pop out? How did you find that?
Yeah, so the way we found that, again, was in the very first pass. We were analyzing these 400,000 reads, and some of the matches that we found in the sequences were to RNA viruses. And this was just really odd. Now, remember, the search that we did was at a protein level. So we're looking, again, at amino acids. And so we found sequences in our DNA metagenome that were clearly very similar to protein sequences that were otherwise only present in RNA viruses. And so this was really bizarre. And at first, we sort of thought, okay, was there some kind of contamination? We're rather skeptical about these kinds of things. And so we actually went back, we looked in other samples, the ones that we hadn't amplified for sequencing, and with PCR we could find this gene to actually be in there. And then we started looking at some of the contig sequences that we were able to make out of some of these sequences. And this was really the work now of Jeff Deemer, my PhD student, who was the lead author on this manuscript. What he found was there was a contig where there was not only this gene encoding this protein that looked like a capsid protein from an RNA virus, was on the same contig as a protein that looked like a replicase protein from circoviruses. These are small single-stranded DNA viruses. In fact, most of them infect vertebrates. And we were also worried, did somehow, when we did the contig assembly, was that incorrect in some way? There's lots of issues with this, particularly in the microbial ecology literature, where things get put together because of the sequences which are similar for whatever reason. So we then went back and did inverse PCR on particularly the capsid protein, and then we're able to amplify the whole circular genome. And I think hopefully TWiV listeners know about inverse PCR. The idea there is, instead of having PCR primers that are facing each other, so you amplify them.
Take the long way around.
And so we did that, and we're actually able to find that these pieces really were on the same piece of DNA. So we'd had this really strange chimera hybrid genome, which looked like it had picked up one piece from the same place as some of these RNA viruses and the other piece from circo-like DNA viruses.
Now, are you thinking that this is in fact a single-stranded genome? And if so, can you find both complements, or is there just one? This is supposedly encapsulated DNA, right?
Supposedly encapsulated DNA. And so we've actually tried, so far unsuccessfully, to show that it is in fact single-stranded, although what we do find in the sequence is a pretty well-conserved stem loop region, which is what's normally required for replication of single-stranded DNA viruses. The rep protein, this replicase protein, which looks like the circovirus rep proteins, mostly seems to work on single-stranded DNA viruses. But again, we haven't actually shown that yet. We're trying pretty hard. I'm beating on my graduate student to do that right now.
Can you find both strands?
I'm not sure I completely follow. So when we did the amplification process, that will actually amplify both strands.
Right,
and
okay.
We don't actually know which one of those was the first one. But by predictions, again, with these stem loop structures, we've got a pretty good idea the strand that should be encapsulated.
Okay.
So how do you know that the virus is actually present in the lake and not just entered it by, say, runoff or an animal urinating in the lake or defecating or something?
Or dying in the lake.
Or dying in the lake, yeah.
We actually don't know at this point, but we've been collecting numerous samples now, and we were just down in Lassen about 3 weeks ago now, and we're in the process of doing the concentration, et cetera, to now be able to look at these in other samples as well.
Yeah.
How many of these bacterial species can you grow to pure culture?
I've got about 50 different isolates in pure culture. Right. And so one of the things we're doing is now using the piece.
Yeah, he's gone.
You're gone. I got to call you back again. We have about 10-minute window of talk.
I think the lake must have exploded.
A dying animal, but if it's there all year round, multiple sampling, probably
Yeah.
not, right?
Exactly.
Right.
Well, we could just discuss this paper without him, right?
We could.
Although he has insights that we don't have.
He
right?
does.
It'd be nice to have him on board.
So if we were to set an experiment up in which we could grow all of the species of bacteria to pure culture, how feasible would it be to use the virus that he's isolated to try to infect any of those cultures?
Yeah, you could try it, sure.
I
I mean,
mean,
if you only have 50, though.
I'm amazed that at 50 degrees centigrade and pH 2.5 that the DNA molecule actually survives that. Why isn't that just chopped
That's the
up?
beauty of a capsid, right? It's protecting it, right?
Yeah,
Rich?
but what about the naked DNA?
50 degrees isn't such a big deal. As a matter of fact, pH 2.5 at 50 degrees might be okay.
Now, are
actually.
these things, do they have very GC-rich genomes to hold them together, or do they use chaperone proteins? Probably a combination of factors,
Yeah,
right?
I don't know.
He's,
We got to get him back.
Yeah, right.
Yeah. Seems like it's hard to maintain contact with the Center for Life in Extreme Environments.
Exactly.
Hi.
Oh,
Aha.
Oh,
we were afraid everything blew up out there.
Look,
Or an
I
earthquake
won't
or,
ask
you know,
any more
other
questions,
volcanic
Ken. I'm
explosion.
sorry.
It was Dickson's question. We're very sorry.
I take it all back.
Yeah. Well, so you've decided what experiments we need to do, right?
Yes,
Yes.
Okay. Good.
that's right.
Well, we did, while we were talking about that, the question came up how these things keep their genomes stabilized
Yeah.
at these temperatures. And is it a lot of chaperone proteins? Is it very GC rich? What's the deal?
That's a great question again. We don't know, to be perfectly honest.
about to go
Turns
offline
out
again.
that they don't seem to have particularly high percent GC. One thing that we have found, at least in the SSV genomes, the ones that we know the most about, there are some DNA binding proteins, and those seem to be important for packaging the genome. At least that's what we thought. And then we used our genetic system to knock out that particular protein in the virus genome, and the virus is perfectly happy. So
Right.
potentially there's a host protein which is coming in and serving the same role as the viral DNA binding protein. We really don't know. We actually just published that a couple months ago.
Okay.
So Dickson wanted to know if you could identify the host by, growing up to 50 hosts in the lake that you can grow and then try infecting them. But you don't actually have virus. You just have sequence, right?
We just have sequence. So we could try doing transformations. And in fact, we're trying to do that right now. Transforming algae is not straightforward.
Another thought, though, was that as you isolate these individual bacterial species and grow them to pure culture, wouldn't at least a small fraction of those populations contain the virus that infects them? And then you might get the virus to actually transmit in the pure culture.
That's what we're hoping. And so we're looking at a number of pure cultures right now to see if that's the case. We're also looking at enrichment cultures because we expect that if there is going to be some kind of virus transfer, potentially before you get to the pure culture stage, and then hopefully find something that's moving around in that particular community.
Right.
So this is a circovirus-like single-stranded DNA genome with a capsid protein coding region from some RNA virus, right?
Yep.
And it's similar to the ones that infect plants, the Tombus viridae, right?
Yeah. So, but that's a relatively weak similarity, and we only really see that. Again, when I say we, I really mean Jeff, my graduate student, who did structural similarity, at least some structural predictions. And at that point, and we again have this in the paper, that you see some very clear similarities at a structural level. At the actual sequence level, particularly for some of these plant viruses, it's really sketchy, kind of on the borderline of something that you would really believe. But again, the structural similarity is pretty strong. The most similar sequences that we found are actually to some plant virus, or I should say some viruses, RNA viruses, which infect mildews. So these are fungal pathogens of some plants.
So one of the other thoughts that I had while I was just listening to all of this is, if I were an ecologist studying the overall ecosystems approach to this lake, these are really ephemeral, unstable environments, basically, in the long term, right? Because volcanoes aren't very stable and they aren't very long-lived. It's amazing that some stability appears to have occurred in an otherwise rapidly fluctuating environment. The amount of water, the temperatures, the pH, all that stuff measured from moment to moment could change on a dime, depending on the volcanic activity underneath it.
Yeah, on a geologic timescale, that's definitely true. We have now about 10 years worth of data on the lake, and there it doesn't seem to change much at all.
Amazing. I mean, that in itself is amazing, because I do a lot of traveling out west. I just got back from a trip, and if you go to Yellowstone Park, and you go to the north entrance, you will see the absence of the Minerva. It's gone. I was there when it was highly active and tremendous to look at, with all these tiers and puddles of different colored hot waters that got all kinds of life growing in it. And one year I came back and it was just dried up.
Yeah,
it had shifted to another part of the Minerva.
Yeah, it's interesting. And some of the volcanic activity, so certainly some of these hot springs, seems to be very ephemeral. This particular lake, however, seems to be pretty stable.
Quite amazing.
Is it stable with respect to the biology, or is there some seasonal variation? I seem to recall a previous show where we talked to somebody who was looking at maybe another environment, or maybe I'm remembering a seminar, I don't know, but and talking about a lot of seasonal variation.
Mm-hmm. So we have done pretty much all of our work in summer. Partly because that's when we have the time to get down there, but also access to the lake in winter is extremely difficult. It's about 17 miles in on a bumpy dirt road under good conditions.
Wow.
And they get a lot of snow in there in winter. So it's been really kind of tough to get in to do much sampling in the winter. But what we did was, and we now being Gordon Wolf's group from Chico State, put in some data loggers actually in the lake itself. And it looks as if over the winter, so they basically went in the very last time they could before they closed the road, put in a data logger, and then came back as soon as they opened the road at the beginning of the season. And what they saw was that, at least at a physical level, just in terms of temperature, pH, etc., it didn't seem to change very much over the winter. You would have occasional excursions getting all the way down to the freezing, cold temperatures of 44 degrees Celsius, and then coming back up actually really quite rapidly.
Now, there are other boiling springs in the world. Obviously, and do you see similarities in the species distribution in them, or are they all? I'm thinking they would be like islands, that they would go through a succession of their own, and you'd see different things emerging in different places.
Yeah, so that's a really interesting area of work, and particularly one of my colleagues, Rachel Whitaker, who's now at University of Illinois Champaign-Urbana, has done a lot of this work with looking at the sulfolobus in some of these springs. And so she's looked basically across the world, because she's using this as an ecological system, looking like islands, exactly as you say, using the island biogeography approach for looking at how these things are related to each other. And she finds very clear differences in the genomes, now using whole genomes of these organisms, from one hot spring to another, and it looks as if there's geographical separation between them. We've done not as extensive work, but similar kinds of comparative genomics with these SSV viruses. And what we find, at least in that case, is there also seems to be a biogeographical signal which is there. That is to say that you have more genetic difference between viruses that you find in, say, Iceland and Yellowstone than you would in viruses between Yellowstone and Lassen.
Okay.
So you also looked elsewhere and you found evidence that there are other related viruses. Is that right?
Yeah. So for this particular case, this one with the RNA virus-like capsid protein and the rep protein. So we looked at the circovirus-like protein. So we've looked actually in a number of different places, and this is all data mining from mostly the Global Ocean Survey data set, which is Craig Venter's attempt to sequence the world.
Right.
And what we found in there were a number of sequences that were actually similar to these RNA-like capsid proteins, again, in DNA metagenomes, and lots of sequences that were similar to rep proteins from circoviruses, but very few cases did we find that they were actually together in same piece. And in fact, I think that's one of the big advantages that we have working in this relatively simple ecosystem that doesn't have so many players in it. We were actually able to assemble a Condit which had both of these pieces on it.
But you did find like two examples in the Global Ocean Survey where you have something that looks, as I understood it, pretty similar to this, where you have actually a contig with a DNA virus rep protein, RNA virus capsid protein. Is that right?
Yeah, so these weren't contigs, they were actually scaffolds. And so I'm not sure again what TWiV listeners know about scaffolds. So basically what these
we
are.
don't even know.
I know what a scaffold is, but that's hanging outside of
yours
Vincent's
paints
window.
a building, right?
That's right.
Exactly. But no, the idea here is there are two sequences that were done on the same piece of DNA, but those sequences actually don't overlap with each other.
I see.
And the way we found these is actually kind of a neat story. So you're looking through this ridiculously large data set from the Gauss survey, and one of the matches we had was to ORF 68,000. Another one is to 68,001. Like, wait a minute. Okay, we've got a rep protein that's similar to 68,001 and the capsid protein that's similar to 68,000. Well, let's go back and look. It turns out that these are two sequences from the same piece of DNA that didn't hook up with each other, but it was the same DNA that was actually being sequenced. So it's one clone, and you just end-sequenced in from that clone.
Okay.
So is that good evidence that these two genes are on the same viral genome, or is that suspect?
I think it's pretty good evidence.
Okay.
Now, we're, of course, doing right now is trying to get more DNA from these sort of environments to see that's actually the case. And I've actually gone back and looked at some other data sets above and beyond what's in Gauss and is present at NCBI. One of the big problems is there's been so much sequencing that's been done, a lot of which is not actually really easily available. So doing the data mining is a little bit of a challenge. There's a group, and I think it's UC San Diego, called Camera, again funded by the Moore Foundation, which has been sort of collecting lots of metagenome sequences. And I went through there and was sort of looking to see, where we had any more similar kinds of sequences. And I found probably my favorite now is one where the sequence of at least the capsid protein was found at 5,600 meters depth in sediment between Spitsbergen and Greenland. About different from Boiling Springs Lake, 50 degrees Celsius, as you could possibly get. So it seems as if this group of viruses is actually really pretty widespread. And
this raises a taxonomic problem. Are you really looking at? I know in your paper you refer to these as RNA DNA hybrid viruses. Is that a classification, or is that just something that happened?
So we are very careful to say that this is a provisional name that we're looking at right now. Again, we just have the genome. We don't have the viruses at all. And so once we actually have the viruses in hand and have an idea what they're actually infecting, then we'll go ahead and actually give these things a proper name according to ICTV, et cetera. And as I think we mentioned in our response to the reviewer, and then let the taxonomic wars rage.
Yes.
Right. Have you?
Yeah, because it occurs to me, this might have happened to some, adenovirus somewhere, and that would not be taxonomically or evolutionarily related to this, but it just involves the transfer of an RNA gene into a DNA virus.
Correct.
Okay.
And so, but then it's sort of, how do you start talking about taxonomy, and how do you start thinking about phylogeny if you have huge amounts of swapping that's going on?
That's why I called it a thorny taxonomic problem. Yes.
So is there any system that you can imagine where you could make use of the sequence of the DNA to actually express the entire virus so that you could get capsid formation and then inclusion of the genome, and then you could find out who's the victim in terms of this infection? Is there a generic system using an archaea that's easily cultured that you could try it out in?
Yeah, there's no nice generic system. That actually sounds a little bit like my next grant proposal.
Lord, if I wrote that one, it would be in Latin and it would be totally unintelligible.
Yeah, so the idea there is really to start to put together some of these things, and what's particularly interesting for us is going to be the actual structure of the capsid protein itself.
Right.
Because that's going to be a really interesting thing in terms of trying to understand really, how different is it? Where did it come from? And so those are kinds of things which we are already working on right now.
So how many proteins does the DNA encode?
So this particular genome clearly has a capsid protein and a replicase protein. There are two more open reading frames that we just call ORF3 and 4,
right?
That may or may not encode protein, right. So we don't know. One of the things that reasons that we'd really like to get some more genomes of these related viruses to see how conserved those particular proteins are.
Right.
Say open reading frames are, because if they're conserved, then they're likely to be something which is protein. If they're not, less likely to be so. So I'm trying very hard to find some whole environments.
So how would a RNA and a DNA virus recombine?
Right.
Bingo. Finally, somebody asked the question.
They did it in the hot tub.
There's the title for this show.
Oh, they did it in the hot tub?
They did it in the hot tub.
It could be.
I like that. Yeah, they did it in the hot tub.
They did it in the hot tub. That's right.
In the deep sediments off of Greenland, as the case may be. They did it.
Yes.
As the case may be. So, but yeah, how this could actually have happened. So we can think of sort of two scenarios on how this actually could have happened. One would be a host that was actually infected simultaneously by a circovirus-like and one of these RNA viruses. And then both of these genomes then have to be in the same cell. And presumably then there's a reverse transcriptase activity, which could be cellular, it could be yet another viral infection. And that then would take the RNA and the DNA. Then the two DNAs could ligate with each other. And then you have your now DNA virus, which has incorporated this RNA piece. So that's one possibility. Another one would be, again, simultaneous infection of an RNA and a DNA virus, but some kind of RNA-DNA ligation that takes place. That then would get copied by the rep protein into just DNA.
Could it be that our thinking about these as being DNA-like or RNA-like, or DNA virus-like or RNA virus-like sequences is wrong, and that rather this represents some sort of really ancient ancestor to both?
So we thought about that, but what we think, because of the actual pretty high similarity between the capsid protein of this new virus genome, or the putative capsid virus protein, I should say, and the rep protein, these are so similar to already extant viruses, we think it's more parsimonious that there was actually from an RNA virus and from a DNA virus. And the main reason for that is we don't see that much sequence differentiation. You'd expect it to something that happened a long time ago. Each of these would really have diverged from each other, and we don't see that when we look at our phylogenies.
And so what's your sense of when this might have happened?
Again, that's the question they asked me at ASM as well.
And what did you say?
We really have no idea. It actually, and I've been thinking a lot about this, partly because of that question, and the fact that we're seeing really similar kinds of viruses in the deep Arctic Ocean sediments and hot springs at 6,000 feet. To me, there are sort of two possibilities there. One is actually did happen really quite a long time ago, and maybe it was the poor animal that fell in the lake or whatever it was, and that it was a relatively recent event, or it could be something which was much more frequent, and for some reason, this particular kind of capsid protein likes to get picked up, or there's something about the virus that encodes that particular protein, which allows it to recombine a lot more. And so again, I think we need to find these particular viruses, and we're also really interested now in the most similar viruses, at least in terms of capsid proteins, these RNA viruses, which are infecting the downy mildews. Unfortunately, these are really not very well-studied viruses at all, but we're hoping that will actually, either our lab or some of the other labs will actually start to look at some of these things, because these may be the real sort of crucial missing links, as it were, between what's going on with these different viruses.
So
and
why
those are fungal viruses, right?
Yeah.
Fungal viruses infecting these downy mildews.
So why can't you take the RNA coding region from your current virus and substitute that for the protein coat from the mildew RNA virus and see what kind of coat you get?
Great idea. Sounds like a sabbatical project.
Hey, you want to go out there, Dickson?
There you go, Vincent.
I've
Now
been
you really
out there.
can
It's
be
a nice.
virologist.
It's a nice town. Portland's a nice
And
place.
Dickson, you could try
you go.
fly fishing in this.
Yeah,
boiling.
they
I
got
know. I've
fish.
been out there. I fished on the Deschutes and the Metolius. It's a great place. It's a wonderful place.
They make beer too.
They do, and I know a nice restaurant. But no,
Yes.
but I, as a non-virologist, I just think about these things like Tinker Toys or Lego, and so why can't you substitute? That sounds like an easy experiment, no?
Well, I think the problem really is with these viruses, the Sclerothorpa macrospora and Plasmapara halsteadii.
I beg your pardon. I thought we were polite people dealing in polite terms here.
Viruses, which are the ones which are most similar. That's how.
Got it.
Like, they really have much in the way of genetic tools. Nothing I've seen in the literature says more than, oh, we've found the sequences of these things.
Yeah, so recovering the virus is the problem.
Even from the mildew, it's a problem.
Yeah, it's hard to do.
I'll be darned.
Yeah.
So we have no picture of an RNA virus capsid from these, I would call them rare viral associations.
Predictions, I guess.
Just predictions.
I'll
Hey.
predict.
I'm going to call you back, Ken.
Yeah.
Should we have him repeat the answer?
No.
No?
To which question?
So
Exactly.
The whole thing. Let's start from the beginning.
Sounds like a preliminary
No,
exam.
just Dickson asked you if you can, substitute the capsid
Oh, right.
coding region, and you said why, but we missed most of it. So why don't you say that again?
Okay, yes. So the main problem with that is that the viruses which have capsid proteins that are most similar to this putative capsid protein that we found in Boiling Springs Lake are these viruses which are infecting downy mildews of plants, and they're very poorly characterized. And so we really like to hopefully either collaborate with people who are working on these or maybe even do some of the work ourselves.
Huh.
Is
Now,
it good?
getting back to kind of this question of how commonly this might have occurred, I mean, that really strikes me that you did find other examples where this sort of thing might have gone on. And looking at the circoviruses and the Tombosphaeridae, it seems like there's sort of kind of a little bit of compatibility there. They're both small icosahedral capsids, and you could picture the circovirus genome would be able to fit inside the Tombosvirus slightly bigger capsid. I don't know. Is that do you think that this would be sort of promoting that sort of exchange?
Maybe I'll try and restate your question. You think that this is a more likely kind of system where this kind of exchange could take place?
Yeah, you're dealing with small genomes and kind of similar-ish looking viruses with just different genetic material.
Yeah, I completely agree with you. I suspect that that's where, if you are going to find something like this is where it would happen. And also, just in terms of thinking about the genes that might be moved from one virus to another, and this is from an evolutionary point of view, if you think about it from the virus's point of view, then picking up a new capsid protein might allow you to have a different host range. It may allow you to escape defenses. And so if there was going to be any protein that would be transferred, I would expect it to be a capsid protein,
Or it might make you more stable in an extreme environment.
for instance.
This is pretty exciting. You have your work cut out for you. There's plenty to do here.
Yeah, there's lots to do.
You didn't address the issue of Lake Vostok. I'm so curious as to what they have found so far, because I know they broke through and they went into the water column, and I don't know what they've pulled up. Maybe you've heard some rumors to that effect.
So what I know about is actually from some of the accreted ice, so before they actually got into the water
okay.
column.
Okay.
And they've found things that look like virus capsids in there. In fact, my second postdoctoral advisor, Mark Young at Montana State University, looked at some of these things in the microscope.
Wow.
But no other information on that at all. And as my postdoctoral advisor said, einmal is keinmal, once is never. Particularly for microscopy, I think that's true.
Right. That could be a title too, Alan, right?
Yeah, that would work.
What was I going to say?
Don't know.
Yes. So this is really good. Another good reason why you should just look in science.
Yeah. Sure.
Right?
Yeah.
You don't know what's
You don't
there.
always have to have hypothesis-driven science, even though in this case you could, obviously you had a hypothesis in the beginning, but just looking is important because you never know what
the basis
you're
for
gonna
hypothesis is observation. I think that's how it starts.
find. Very good, Dickson. I like that.
Thank you.
Do you use that for the NIH or?
I used to. Eventually, they didn't buy that. They called them fishing expeditions.
Yeah, well, in this case, it's fishing with a big pole.
I told them I was very good at that, and they didn't believe me.
One of the things that I was actually struck by again in thinking about this TWiV was a quote which has always been attributed to Pasteur. Chance favors the prepared mind, or something like that. But I'd never found the actual French until I went and looked for it. And do any of you know it?
No.
Luck favors the prepared mind is the way I know it.
Yeah, so dans le champ d'observation, le hasard ne favorise que les esprits préparés.
Wow.
Which actually, I don't like any of the translations, and I'm not a very good at translating myself. But it sort of a couple of things in there is that it's talking about observation, and so really thinking about an observational approach to science. And basically, it's a classic kind of French in saying, well, basically, it will only hit you over the head if you're ready for it to hit you over the head.
That's right. That's right.
And that ties in actually really nicely to basically our paper. And one of the reasons I mentioned right at the beginning is why we wanted to publish in Biology Direct and get it out quickly. Because there are a number of publications that actually have tables in them, in, again, journals that will remain nameless, which actually have RNA hits in a DNA metagenome for a virus metagenome, published well before we even published ours.
And they didn't pick it up, right?
I guess there's esprit were not as préparés as ours were.
There you go.
Perfect.
Très bon.
Well, I suspect that this paper is going to generate a lot of interest, and people are going to be looking for these a lot more because you found them.
Yeah, we definitely hope so.
It's cool. All right.
Very cool.
Anything else before we move on, gentlemen? Okay. I think we should move to email and save. We had a couple of other papers here, but we'll save them to next week. There's always next week.
There is.
First of all, there is one note here. Another TWiV bump has been noted. Kathy Spindler has a PhD student, Qian Hui, who will be doing a postdoc with Matt Evans, and she heard about the opportunity on TWiV.
Nice.
Because we mentioned that Matt had postdocs available.
Nice.
She listens to TWiV.
How about that?
Bingo.
Luck favored the prepared ear.
Really? All right. Now, we had an email that Rich and I read on TWiV 193 and ASV, and Rich, you wanted to revisit this, right?
Yeah, because although we addressed some parts of it, we never addressed sort of her major question, which is, I'll reread one paragraph of this. What drove me to write today is because I was trawling through the literature trying to find a journal article. This is Dina writing. Trying to find a journal article that would entice enough people to attend our voluntary journal club and hopefully stimulate an interesting discussion. We do all right for a department of 35 postdocs and PhD students. We provide pizza and drinks and usually keep about 10 participants entertained for an hour. Our journal club is run in the traditional way. There's a presenter who reads the original research article thoroughly and uses PowerPoint slides to present figures as he or she explains them. We ask questions during the presentation and have a general discussion at the end, but there is a little voice inside me that says, this could be better. Therefore, I would like to ask you, the TWiV hosts, and your listeners to share your experiences of journal clubs or other brainstorming / discussion / mixtures that you have tried, and what you have found that makes the experience a little bit extra special. So I think, we never really discussed that, and I think it'd be worthwhile. I can start with 2 things. First of all, although nowadays there's probably no way around it, one of the things that stands out to me here is the word PowerPoint.
Right.
Yes.
Which, it's very useful and that kind of stuff, but it also, in particular, if you're not very good at using it, has the potential to just really make everybody shut down.
Yeah.
Okay?
Mm-hmm. Yeah.
Just put you right to sleep. So you have to be really careful with that. The other thing, we run a virology journal club here in the spring where now there are students enrolled for this and they're getting credits so that we can actually ask them to do stuff. And what we ask people to do is to genuinely have everybody read the paper before the journal club, and to enforce that, the presenter doesn't actually present the whole thing. They present an introduction or whatever, and with each figure, ask for a volunteer from the audience to go over the figure. And that really compels everybody to do a more thorough job of preparing for it. And the more people that are actually prepared for it, and the more people who are actually talking during the presentation, the more engaged everybody becomes. And what we find is that by the halfway through the semester or so, the group really is sort of more into it than in most other journal clubs that I've participated in. And it usually winds up by the end of the semester that we have really pretty good group discussions every time. It gets to be more like a mega TWiV than a journal club.
Yeah, I certainly recall, Vincent, there was the advanced topics course. I don't know if I don't think it's still structured the same way, but in graduate school, this was a major thing, was one of the big courses in second year. You'd sit around a table with a faculty member and you were assigned a paper to read. And it's very much like what Rich is describing, except it wasn't asked for volunteers to describe a paper. It was, Alan, walk us through figure 3.
You had to do that. That's right.
And people, you'd get into kind of a rhythm where sometimes you'd be counting ahead. Okay, now I'm going to be doing figure 5. Okay, I better really bone up on this. But that kind of thing will really focus people's minds, let's say.
Yeah. I ran a course once. This was years ago in Buffalo, where I didn't want to give exams or anything, and this was one of these literature-driven courses. And what I told the class was, okay, there's the way we're going to do this. There's going to be oral exams. That's how you'll be graded, and these are going to be short oral exams, and they can happen at any time during the course. And each of you is going to get 4, and I'll use 3 of them. And what it consists of is that any time during the discussion, I'll just stop the discussion and say, so-and-so, you're having an exam. And I'll have a conversation with you. And as long as you can talk intelligently about what's going on, then you pass the exam. And I'll tell you, everybody was awake for that sucker.
So, Rich, have you managed to...
And by the way, the snakes are very real.
Have you managed to scrape all the tar and feathers off your body, Rich, since you moved to Florida?
Once everybody got used to it, it was fine.
Oh, of course.
Yeah, I would reiterate what you said, Rich, about PowerPoint. The journal clubs that I remember being really successful were chalk talks.
Yeah.
Mm-hmm. Yeah, I would agree with that also wholeheartedly. The less technology, the better.
The one of the problems is that you do want to project figures and stuff, so that you could probably structure this so that you could say, okay, you can use PowerPoint, but there can't be any words. Well, I mean, you'll only use PowerPoint to actually show a figure so that you can point to it.
Right. You can use PowerPoint for figures only.
Right. No text.
My pick of the week, I'm going to get one here, is Edward Tufte's little handout, which you have to actually purchase for a minimal sum, on the do's and don'ts of PowerPoint presentations. And it's words are out. You're absolutely right, Rich. Words are out. No bullets, no sentences, nothing. Your visuals there, and the handouts are the words.
It's funny. I may have mentioned this at TWiV ASV, Rich, but I was live tweeting some of the talks in the morning, And to do that, I'm just looking at my keyboard and not at the figures. And I found that I could understand far better if I wasn't looking at the figures. I didn't even need to look at them. For the good speakers, you could get everything from just listening to them.
Yeah.
And, and I think in a talk like that, a plenary talk, you just need the big picture, right?
Well, actually, you make a really good point here, because I am constantly trying to figure out for myself, what is it that makes a really good talk relative to one that's just not so good? What is it that really engages me in a good talk? And I think you're right. It's the actual speaking. It's the voice and what they're saying, and it doesn't have anything. It doesn't have much to do necessarily with the visuals.
No, you're quite
So I think in a journal club, if you can work it without figures as much as you can, it really helps.
And also to make everyone participate, they have to want them to participate, I think. I can only rely on my prior experiences, of course, and the best course I ever took in my whole life was an American literature course as an undergraduate. And if you didn't read the book, you had a week to read these books. If you didn't read that book, you felt so left out of that class. You didn't know where to go. And the guy who taught it made it so much fun that you wanted to participate, because you got a chance to tell a joke, or you got a chance to show off your knowledge or stuff like this, and that's what he really wanted you to feel like doing. And he engaged every single student in the audience, and that's the trick, is to make eye contact with virtually everybody and pretend that they're the only ones in the room. And if you do that, you'll have a much wider audience. And a lot of these journal clubs are done because they have to. It's a 1 credit, every student has to show up, and it's just a drudgery. Just off. Pizza doesn't do the trick here. Right?
And by the way, when you say pizza and drinks, I assume you mean beer?
Right. Not allowed on most campuses.
That might help too.
Most campuses won't let that
here on
happen.
TWiV, we don't use visuals for the most part.
No, we don't.
And people do get it.
No, I think radio versus television has a great discrepancy of opinion. You learn much more listening than you do looking. I think you do, even though your optic lobes are bigger than all the other parts of your brain.
I think it depends on the material, but it is good in an audio format. You've got a projection screen that's as big as the listener's imagination.
There you go. That's the best thing I've heard today. That's terrific, Alan.
How's that, Rich? Good?
Great. I really like the comment that TWiV is without visuals, because that makes perfect sense.
And
so that's good. We did that justice.
If anybody has other ideas, send them in.
Yeah, because this is it's a common, it's a perennial problem, and so everybody will benefit from people's ideas on how to deal with this.
I personally feel that the discussion approach should really get more traction at meetings. The typical science meeting, you go up and you put a PowerPoint. I think it can be numbing. Yeah. I think you put 4 people up front and you have them do a TWiV-like discussion.
A panel.
And you can have the audience ask questions.
Sure.
I think it's much, much more compelling.
Well, I think that's why poster sessions are so popular. You get a one-on-one with the person.
Yeah.
That's our 5 cents, right?
That's right.
More like $1.50. Yeah.
All right, Alan, would you like to take Ben's email?
All right, this one's I think we're going to be on this one for a while, so this will probably be the last letter we do for the show. A great letter, by the way. So Ben writes, "Hello, TWiV crew. I must first apologize. It's been a long time since I've listened to the podcast, but I promise to get caught up. I'm seeking guidance from you as fellow scientists here. On a matter of personal and professional development, I was curious as to the most effective and sanity-preserving ways you deal with pseudoscience, or the complete lack of acceptance of science and evidence-based medicine by some people. A little background. I'm entering graduate school going for a PhD, and when I try to have discussions with my non-science friends or acquaintances, I hear a lot of science bashing and evidence-based medicine bashing. I know this has been experienced by you all in the show many times, XMRV and chronic fatigue being an obvious example. On a personal level, I've experienced it myself, but in a strange way, a sort of reversal of the normal paradigm. I have ADHD, attention deficit hyperactivity disorder. I know it's a controversial issue, but I have it, and I've been prescribed by my physician for some 15 plus years now a medication to help me focus. It's worked for me wonderfully. I did well in high school, excelled in sports, have had great work success, got my BS in biology, performed independent research as an undergraduate, and I'm now going to be working on my PhD. Yet I have people out there that are telling me, I don't have a disease, I don't need to be medicated, despite the fact that it works and I'm successful. I just need more quote-unquote fresh air and to exercise and eat right and get more vitamin D. And I see posters like, childhood is not a disease, don't medicate children. I try to have rational science and evidence-based conversations with these people, but I'm just a crony of Western medicine, or I'm only treating the symptoms, not the cause, etc. I worry that a child who's in my situation may not get the help I got. I just don't know how to deal with people like that who refuse to accept evidence or only accept anecdote when it works for what they believe. Not that anecdote is ever a good argument. I'm a man of truth, that's why I love science. It upsets me when people don't want to hear evidence or don't accept the scientific method. I tell them questioning is good, but you have to also examine evidence, not anecdote. I truly want to educate people and greatly enjoy it, but I'm saddened by people who refuse to learn and worry that people like that could run our country, and in turn try to control my life based on unscientific beliefs.
You mean do run our country?
Yes, I think that
Rich, you took the words right out of my mouth.
Unfortunately true.
Or have been running our country.
Someone is honest enough to tell me they didn't know something and that I taught them something and changed their mind. And it felt really good, hence why I want to be in an academic setting. How have you learned to deal with people like that? People that refuse to learn. I don't give up on them, but is that necessary? Should I just pick my battles? I know your podcast has been extremely successful in educating people, especially outside the academic setting. Do you feel like the battle for knowledge and education is being won? Sometimes I actually become saddened that people are not willing to learn or accept evidence, but I don't want to let that ruin my career seeking truth. I thank you for reading this. I know you're all very busy. I don't expect this to be read on the air. There you go.
That's right.
Did you see fit? We didn't. Sincerely hope for a response of some kind, time permitting, via email, hopefully maybe a few words from each of you, since you all may have different opinions or strategies that have helped you to success.
Okay. One should never send an email to TWiV figuring it won't be read on air.
That's the kiss of death. We'll read it every time.
Eventually we will, yeah.
Sure. It's like I once had a conversation with potential astronauts, and I said, what made you become an astronaut, since I must assume that not everybody gets to go up? And he looked at me astonishedly, and he said, but everybody gets to go up. I said, really? That's incredible. So what do you do about somebody who doesn't believe what they see and what they hear? That's the question, I think.
To me, the key phrase in this letter is pick your battles.
Yeah.
I think there's only so much you can do, and then you start driving yourself crazy. There are some people who are simply have their minds made up and they're not going to listen. It's not as if you shouldn't, explain your perspective to them, but after a certain point, there's no more you can do, and you simply frustrate yourself. You do what you can, but there's a limit to what you can do. So pick your battles, and occasionally you're gonna make a little bit of headway, and that's good. But don't expect much.
I used to tell my kids, you can lead a horse to water, but you can't make them think.
Right.
Yeah. That's just Ken here. I just wanted to make a quick comment and to some extent a plug for the video that we you mentioned really earlier on in TWiV, and that is to think about having other people talk about your research. And so the video, which hopefully we can get linked on the website, is about a independent filmmaker who knows nothing about virology, but is actually doing a documentary on viruses and sort of how viruses may actually be able to be used for good as opposed to just making us sick all the time. And I think that reaching out to some of the people who have a very different approach of doing things, again, in this case, a professional filmmaker, can potentially have a lot of influence. Again, don't know. We're still in the process of making it, but hopefully that will help.
Well, some people don't even believe. There's a huge controversy with regards to climate change issues, and that whether we're responsible or not for it, the climate is changing, and people don't even believe that, and it's right before their very eyes. They can see it, but they don't believe it because they don't want to. So if you encounter people that don't want to believe anything because they have their minds set already, that's a lost battle for them, not you. You want the ones that are coming in prepared to learn. And that's what the college situation is about.
Yeah, and I find this is actually much easier to deal with one-on-one in person. People tend to be a little less extreme in their convictions when they're actually talking to a person who is reasoning with them. Yeah. If you get drawn into debates on the internet, sorts of folks, you're never going to get anybody to budge.
No, that's true.
So, state your position, make it clear that you're going with the evidence, and then just leave it.
Right.
But yeah, pick your battles is really, really the key to this, because this is an ongoing frustration for everybody, not just everybody in science, but everybody who follows science.
Sure.
All right, here's my approach, if anyone cares.
I care.
We all care.
So I don't do TWiV to convert anybody, although we might one or two along the way. But I'm doing TWiV to get to people who will someday be teachers and can teach people what science is and why it's important. Mm-hmm. And I teach a virology class to undergraduates, and I tell them in the first lecture, I'm teaching you this because you're going to go out, you're going to be teachers, journalists, policymakers, politicians, and you're going to know about viruses and science and why it's good. And you can influence, each of you can influence two people, and they can influence two more. So it's the same thing, a little sort of picking battles, but I think the key is education of very young kids and getting them to appreciate what science is. And that's going to take a long time, but eventually if we all do something, I think it can help.
A big influence is a person's parents and what they believe, because they're the first ones you encounter before you get to the school systems. And we've all encountered this, I'm sure, along the way of our maturation process. Our parents may have had certain beliefs that didn't correspond to what we were hearing in school, and you'd come home and you'd run that by them, and they would just say, well, that's not true because, the Bible says it, or because I heard it from my grandfather, or I know the Mississippi River is red because I was told that by somebody. And when you present your own parents with the truth and they still don't accept it, that's that's grounds for divorce at that point, and I know lots of people that have gone through that by choosing careers in science where their parents were not science people. And, it's a hard deal. It's really a hard deal, but you have to stick to your guns.
Anyway, it's a great email.
Yeah.
Thanks, Ben.
Yeah,
open-ended answers. We will love answers from our listeners.
Sure. Always. TWiV at twiv.tv, right, Dickson?
Got it.
Let's do a couple of picks of the week. Dickson, you wanted to pick.
I did, but I forgot. I think it was a paper on Alzheimer's.
Alan put it in. Why don't you tell him what he's picking, Alan?
Oh, wait, yeah.
What did I pick?
Tell him what he wins.
Tell me what was behind that door, Alan.
Dickson, you have chosen Edward Tufts, The Cognitive Style of PowerPoint.
Yes!
Checking out Corrupts Within.
That's
available for $7 from Tufts' website.
Yeah, $7. That's too expensive. Much too expensive.
Should be $1.99, right?
The gist of that is don't use words in your PowerPoint. Use pictures. And say the words.
Alan, what do you have for us?
I have a collection of pictures. These are scanning electron micrographs of insects.
I like visuals.
These are terrific.
I love them.
And these are color scanning.
Colorized.
So they've been colorized, and they're really, really cool.
Who did them? Do you know the origin of them?
These are from
Steve.
Yeah, right.
Pronounce that.
Schmeissner.
Okay.
His website. I dug a little further into this. His website is a trip in itself.
Yes. Yeah, actually, we should probably link to that as well, because it was posted on one of these sort of meta. Here are some links you might like.
Right.
Blogs. Cool. But this site, theworldcloseup.com, is this fellow's collection of really, really cool images.
Nice.
So did you notice, Alan, unless I'm mistaken here, some of these insects have their own little parasites?
Oh, of course.
Look down at the yellow dunfly.
Mites. They have mites.
See him? He's got little mites or something around.
Oh, absolutely. That's what's killing off the bees.
Wow.
Yeah, the technology for a lot of this SEM is also getting really advanced.
Yes.
And one of the things that a local microscopy company here, FAI, in Portland, or actually just outside of Portland, they actually now have a scanning electron microscope designed for high school students. Wow. It's about the size of a PC. Wow. It's just amazing.
That's incredible.
That's nice.
And it's really wonderful. And one of the things that we were looking at, just brought up by this, they had a Drosophila which they'd mounted onto an SEM stub to take a look at. And just in looking at the Drosophila, I could see that particular Drosophila had been sitting around for a little bit too long. You could see it clearly had a bacterial infection around its eyes.
Wow.
This is just fabulous.
Nice.
And these are kinds of things. They're really designed for high school students. They really want to have SEMs in high schools.
It's a great idea. When they can see this stuff, they get really turned on.
Yeah, they sure do.
Yeah, that's great.
They sure do.
Rich, what do you have for us?
Well, Vincent and I have very closely related, partially overlapping picks because both of us obviously got off on the Mars landing this week.
Dickson, you were listening.
Of course.
Did you hear what Dickson said, Rich?
What do you say?
He called it a contig.
Very good.
Yeah.
As most people know, there's a new robot that was landed on Mars this week. And when I looked at the descriptions, I hadn't really followed this. When I looked in the descriptions of the newspaper as to how this thing actually landed, they talked about cables and stuff, and I couldn't figure it out. So I went looking for I figured there's got to be a video somewhere. And I found a video. There is an animation that has been put together of the landing sequence that is shows up in different videos in different formats. The one that, and I'll let Vincent describe the sequence if he wants, but the one that I showed, the one that I found is kind of interesting because it interlaces the animated landing sequence with the reactions of the control people at Jet Propulsion's laboratory as they're getting the news of the events and the landing. Right. And it's really awesome to see this group of people. You feel the tension as they're anticipating whether or not this thing is going to work, and then the release and the excitement when it does work, and it gives you a really unique flavor of the sort of the intensity of the excitement of the
amazing accomplishment.
It's really
just
really
an amazing
something.
Accomplishment.
So that's a nice video. Vincent's got another one.
Yeah, so my video is also from NASA. It's called Curiosity's 7 Minutes of Terror. And this is a very nice interspersing of interviews with NASA people who are working on this mission and animations of the landing. And it is so dramatically done. I mean, they got really good people to make this video. It is great. It's basically an interview, but it's so well done.
It's so well produced. It's just
great.
Gorgeous.
And you go through the sequence of events that this lander has to go through. It's coming towards the planet in a capsule. It's going too fast, so they put a parachute out, but that's still too fast, so they have to slow it down with rockets.
Right.
Yeah.
But they can't land it with the rockets because it'll blow dust over everything. Exactly right. And so all of this, and then the cables, they lower it on cables, and then they, at the last minute, they drop it and the rocket goes away. It's just really well done. So
can you believe that all worked?
It's amazing, yeah.
It's astonishing.
Incredible engineering.
That's what they said. They couldn't believe that it worked. They said, this is just too complicated. We have all the pieces together. We know that they all checked out on Earth, but it just is impossible, and then it worked.
So in one of the other ones, they let it bounce on the planet, right?
Yeah, they had balloons.
Yeah, they had it encased in balloons.
But did you see yesterday on their news show, they actually photographed with the Mars orbiter, the reconnaissance orbiter, they photographed the parachute opening.
Yeah.
Yeah. That was awesome.
Remarkable.
Yeah, so this is this whole program of Mars landings, about $2.5 billion, which is really nothing for what we're getting here. And this is just, this is why we have to spend money on science, man.
I found myself walking and thinking about this the other day, thinking, before I've been wondering, do we really need to send people to Mars? And now I'm, I'll go.
Why do we have to send people? We got these things.
I know, but just for kicks.
For kicks?
Yeah.
Hey, there might be some aliens there. You just don't know.
Yeah, we put them
there'll be viruses there. We have to find the viruses there.
No, we've already contaminated the planet. We're going to find our own microbes there, I'm afraid.
Yeah, that's true.
Ken, you must know Forrest Rower, right?
Oh, yes. Yeah, actually, I stayed in his house last weekend.
So on yesterday, I do another podcast called TWiV, and we discussed some of his work. And he's talking about, he's a big viral metagenomics guy, of course. And he's talking about all the novel phage-encoded genes that have yet to be discovered on Earth. There are 2.5 billion of them. Mm-hmm. Okay. And then he says, I sincerely hope that there are even more on Europa, which is someplace where there could be life, right?
Yeah. Mm-hmm.
So that's pretty cool.
That's, again, I don't want to be shamelessly plugging the video, but that's, in fact, one of the things that we talk about is, landing on Europa and, are there viruses there?
Yeah. Now, we'll definitely link to that video. Is that you in the video taking a sample of water?
That's me taking a sample of water from Boiling Springs Lake, in fact, the place where we found these the new virus genomes.
And the actual movie is going to be a full-length, what is it, a documentary type movie?
That's the plan, yeah. So we're planning on having at least 1 hour-long documentary, and the idea is really just sort of an educational, but for a much broader audience kind of thing. Right. And at this point, we're frantically trying to scrape together enough to get everything all finished. And we got this trailer going, and we're hoping to keep moving along with it.
Wow, good luck.
So, Ken, did you want to pick something? You want to make that your pick of the week, that trailer?
I think I'll make that the pick of the week, but I also wanted to mention that we also have a Facebook page. Which discusses some of this as some of the outtakes from the movie, some more pictures of what we're doing, also posts to TWiV and things like that, which are going on there. And it's really easy to find if you just go to Edge of Life, the movie, just all one big word. I guess it's a contig. edgeoflifethemovie.com. And so that'll be my pick of the week.
All right. I'm logging into Facebook right now.
I got it.
So and if you like it, please like us.
Yeah,
you are now liked.
I got it. I'm liking
Absolutely.
It too. So we have
cool pictures.
We have quite a few fans on our TWiV Facebook page, so they'll all see it as we like it. So we got
excellent. Nice pictures.
Yeah, cool.
I particularly like the one. In fact, I think my last posting on there was the RAU student research opportunities for undergraduates. Is then behind the sign that says illegal to go beyond this point. She just presented her final RAU presentation this morning. Yeah,
got it.
Wait, there's a location in this park called Bump Ass Hell.
Yes,
Bump as Hell. Yeah.
Mm-hmm. And the reason for it's called Bump as Hell is the reason that we don't go sampling in winter. That's because poor Mr. Bumpus, when he went into Bumpus Hell, fell through the snow and lost both of his legs.
He lost both of his legs.
His legs. So presumably what's above the legs is still there. But
oh dear.
I like this quote from William S. Burroughs. Language is a virus from outer space.
Oh yeah,
I didn't know he said that.
Did you ever read anything by William Burroughs?
No.
The Last Exit from Brooklyn is very interesting indeed.
Is that your pick?
No, that would not be my pick, actually.
Thank you. All right, we have a listener pick of the week. We have two. One is from Lance, our friend at the Wellcome Trust. The first is a website called vaccineconfidence.org. It's the Vaccine Confidence Project monitoring public confidence in immunization programs. A lot of cool information about what's going on in that area. Very cool. So check that out. And then we also have a YouTube video, which is an introduction to immunology.
Oh.
And
it's one of the weirdest videos I've ever seen. But have a look.
Have a look. I'm not going to play it now because everyone will hear it. Still enjoying the podcast, he writes. Keep it up. Still. Hell.
Still, wow.
Really. And we also have a pick from Anat, who says, Hi, TWivers. My name is Anat, a PhD student working on human T lymphotropic virus at Imperial College London, longtime TWiV listener. I would like to suggest the Beautiful Science Project as a potential listener pick. Beautiful Science is a project that currently involves around 30 participants. It invites artists and Imperial College biomedical scientists working on cutting-edge research to collaborate and create artistic interpretations of the scientists' work. The website, beautifulscience.info, details the progress of the project. And for those in the UK, there will be an exhibition of the artwork at the Brick Lane Gallery in London between 26 June and 2nd July. Well, I'm sorry.
Whoops.
We missed it. I hope it's okay to suggest a pick that is organized by some of my colleagues, but I thought it might be of interest to other listeners.
It would have been if we'd gotten to this.
This is true.
Sorry, it's my fault. I should look at dates. Anyway, you can still visit the website.
Right.
Thank you for a wonderful podcast. Okay, that'll do it for TWiV 195. You can find us on iTunes, the Zune Marketplace, and TWiV.tv. If you do use iTunes, you should subscribe to us and leave a comment or rate us. That helps us stay very visible in the iTunes area, and more people, therefore, will find us. And that's what we want. We want to spread the word about viruses. We also have a Facebook page, facebook.com/thisweekinvirology, all one word. And as I said before, send your questions and comments to TWiV at twiv.tv. Dickson Despommier, good to see you again.
Vincent, it's a pleasure to be back on track.
On track. You can be found at verticalfarm.com, medicalecology.com, and Racaniello.org.
Yes.
And you're a blogger, too.
I am a blogger.
At verticalfarm.com, right?
This is true. I have to go write one now.
Excellent. Sorry to take you away from your
No.
task.
No, I wouldn't miss this.
Rich Condit's at the University of Florida, Gainesville. Thank you, Rich.
You're quite welcome. Always a good time.
This was very much fun today.
Yeah, good fun. Good
And
fun.
by the way, in the time we have spoken, the clouds have gone away, the sun is out, and the temperature has gone up 2 degrees Celsius.
There you go.
We're at 23 degrees. Alan Dove is at alandove.com. Thank you, Alan.
Thank you. Always a pleasure.
We'll see both of you in a couple of weeks, won't we?
Yes.
Yeah.
But we're not telling anybody what it is, but it's for the 200th episode.
Yep.
I get to meet Alan Dove
In person.
in person.
That's right. And our special guest today from the Center for Life in Extreme Environments, Ken Stedman. Thank you, Ken.
Thank you guys for having me on. It was a lot of fun, and the sun has come out here.
Hey.
And it's about 4 degrees warmer.
Amazing.
Bi-coastal. Go outside and enjoy it.
I'll do my best. It's almost lunchtime, so
Oh, I forgot. You're in the middle of the day here.
That's right.
We're wrapping it up.
Give my regards to Peter Platt at Andina, please.
I'll do my best. Yeah, they have very good food.
They do.
Yeah.
I'm Vincent Racaniello. You can find me at my blog, virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.